Okay, in this video, I'm going to cover the beginning of the immune system. So when we're thinking about the immune system, we can pick up from what we learned at GCSE in that we can think about what we call the first stage of defense. The first stage of defense is the idea that we try and stop the pathogens from getting into our bodies in the first place. So for example, mucus production in the various parts of the body, which are going to trap your pathogens. Or the skin and blood clots or scabs, which act as a barrier. You can also think about stomach acid, which obviously kills the pathogen. Okay, it will digest it. Now we know that some pathogens are capable of getting past the stomach acid, but we'll leave that for the moment. We have cilia, which waft the mucus, waft the pathogens in the mucus. So we're talking about cilia within the trachea and within the bronchi. So this is our first stage of defense or first line of defense. Okay, stop them getting in. But what I'm really interested in is the next line of defense, the second one. So the second stage of defense is our non-specific immune response. In other words, it will attack any particular pathogen. And here we are dealing with the process of phagocytosis. So if we first of all just draw a little picture of phagocytosis, we'll have our phagocyte. Now you'll notice about our phagocyte, it's got this lobed nucleus. It's obviously got a very flexible membrane. And then we'll have our pathogen over here. And the pathogen does actually secrete chemicals which the phagocyte is attracted to. What will happen is the phagocyte will come along and it will invaginate its membrane, which means it bends it round, so that the pathogen starts to be taken in by the process of endocytosis, like so, until eventually we have the pathogen completely inside our phagocyte. It's completely been engulfed. And it's sitting inside something called a vacuole or a vesicle. You can call it either, it doesn't really matter, both are fine. What will happen next is to do with the structures called lysosomes, which we know contain hydrolytic enzymes. And they will fuse with the vesicle or vacuole and release the hydrolytic enzymes into the actual vacuole. And therefore, what they will do is they will hydrolyze the actual pathogen. Now, note I use the word hydrolyze and not break down or digest, until finally the antigens, which are the foreign molecules found on the surface of our pathogen, the antigens get displayed on the surface of the phagocyte. And we now call this cell an antigen presenting cell. So let's think about what I would do if I wanted to describe this in words. So if I wanted to describe it in words, I would go through the number of stages. So the first thing I would say is that the phagocyte is attracted to the pathogen by chemicals, and it engulfs the pathogen. Pathogen is placed inside a vacuole, and the lysosome fuses with the vacuole, releasing hydrolytic enzymes. hydrolytic enzymes hydrolyze the pathogen, and the antigens are placed on the phagocyte's cell surface membrane, forming an antigen presenting cell. And obviously that's quite a long description, so it depends on exactly what they're asking in the question. Okay, so that's the first stage. So there's a couple of things that we obviously mentioned there that's really important that we can actually describe. So we've got the word pathogen, which we talked about quite a lot, okay, which you should know from GCSE, a disease-causing microorganism. And then you have antigen, which is a foreign molecule found on the surface of the pathogen or of the cell, because it could be a foreign cell. That causes an immune response. Normally a protein, but in theory, it could be something else. Okay, so we now lead to the second stage, the third stage, my apologies. We now lead to the third stage of defense. Now the third stage, or third line, as it's sometimes called, is our specific immune system. And the specific immune system does rely on the non-specific immune one, because we're going to see that it does rely on the actual antigen presenting cell. But we're talking basically about three cells here. We're talking about T helper, T cytotoxic, and B cells. And T cytotoxic is what we call your cell-mediated response, because it's going to be the cells that are actually going to attack the pathogen. And B cells is called your humoral response. But what we shall see, to summarize first, is what's going to happen is, first of all, you have phagocytosis. And phagocytosis makes antigen-presenting cells, APCs. The APCs activate the T helper cells. And the T helper cells do one of two things. They either activate the TC cells, or they activate the B cells. And in fact, they'll do both of those things. And they'll do them concurrently. So first of all, you have to have phagocytosis. You have to make your antigen-presenting cells. Your antigen-presenting cells will activate your T helper cells, and then your T helper cells will activate your TC and your B cells. Now, in an exam question, often you're just asked about B cells. Okay, and when we're just asked about B cells, we're going to only talk about the activation of the B cells. If we're asked just about the humoral response, we're just going to talk about the B cells. But sometimes you might be asked about the role of T cells and B cells, and then you're going to go all the way from there. Obviously, to understand it, we're going to go away from this process. So we need to think about how we activate our T helper cell. So we know that we have just made an antigen presenting cell, and that antigen presenting cell will have on its surface a very specifically shaped antigen. And we know that there are a variety of T helper cells. We'll put them in green. There are a variety of T helper cells around that have different receptors on their surface. And there are different shapes. And only the T helper cell with the correct shaped receptor, i.e. the complementary receptor, will be able to bind. So the T helper cell that has the complementary receptor will bind to the antigen presenting cell, and the antigen presenting cell will then cause that T helper cell to clone itself. So that clone means that they're going to produce lots and lots of copies by mitosis. This is often called clonal expansion. And those copies, which are all identical to each other, because they've been produced in mitosis, so they all have exactly the same receptors on their surface. I'll just draw one of each. They will then split up into two types of cells, some that are active and some that are memory, and the memory ones will remain for future infections. So once again, if we want to just describe this in words, okay, we start by saying a specific T helper cell binds to the APC in what they call clonal selection. It's a very useful term to use. It's often referred to in the mark scheme. The T helper cell is activated to divide by mitosis in clonal expansion. Again, another good term to use for the exam questions. Some of the T helper form memory cells, and the others form active T helper cells. You can just call T helper cells T small h. Okay, and that's absolutely fine. So that's the first bit. Now the second bit we said was that those T helper cells have one of two jobs. So I'm going to deal firstly with the T helper cells that are going to activate the TC cells, because you don't really need to know much about these, but the B cells we need to go into a little bit more detail about. So the job of the T helper cell is that some of the T helper cells, some of the active T helper cells, so I just write it as TH, will then activate TC cells, which are our cytotoxic T cells. Okay, and again, this is going to be a clonal selection because it's a specific one, followed by clonal expansion again. We've got exactly the same process going on, but we're activating our TC cells. Now, the job of TC cells is to kill infected cells. That's all they do. They normally punch holes in the membrane so that the contents of the cell leak out, so the cells get killed. There's not much more to say, okay, about TC or T cytotoxic or T killer cells. It's all the same thing. So they're activated by clonal expansion and then clonal selection, then clonal expansion, and the T cells will kill the infected cell. I want to concentrate on the second one, which is the activation of B cells, because that's the one they tend to ask about the most. So imagine we have our pathogen with its antigens on its surface like that. Okay, what will happen is our T helper cell that we made up there, our active T helper cell, that will bind because it's got the receptor that is complementary. And then once again, we've got lots of different B cells out there with different antibodies on its surface, but the one that has the correct antibody will bind, so the specific B cell. The T helper cell is then responsible for activating that B cell. And again, that B cell will then divide by mitosis, clonal expansion, to give lots of identical B cells that can either form the memory B cells or they will differentiate into plasma cells, and the plasma cells will then secrete the antibodies. So again, how do we describe this in words? Because that's what always the difficult part is. Okay, so the first thing you say is that the specific B cell and T helper cell bind to the antigen. The T helper cell activates specific B cell in clonal selection. This causes the B cell to divide by mitosis in clonal expansion. Some of the B cells remain as memory cells. The rest differentiate into plasma cells that secrete antibodies. Okay, so let's think about what an antibody actually looks like, because we do need to know that. So you should know that an antibody is a Y-shaped molecule. Looks like this. It's made up of 4 chains. Okay, so we've got what we call the heavy chains, which are the long ones. And then we've got the light chains, which are the ones that are there. They have bonds which hold them together at various points. Okay, those are called the disulfide bridge. And this area over there is called the hinge because it allows the antibody to open and close. So if you can imagine, the antibody could close like this, or it could open like that. Okay, so it allows the different shape. And then when you're looking at the antibody, the binding site for the antigen is always on the end. And that is because this area over here is called the variable region. That means it changes within each antibody, whereas from there onwards is the constant region, in that it's the same in every antibody. And antibodies generally do one of 2 things. They either neutralize, which means that they stop the pathogen causing harm by entering cells, or if they're antitoxins, then they will bind to the toxin, okay, and stop that from causing harm. Or they're what we cause agglutination. To agglutinate means to stick. So it clumps the pathogens together. And when they clump the pathogens together, they can then be engulfed in phagocytosis more easily. So I'll draw it and show what I mean. Not the engulfing bit, but the clumping bit. So if you imagine you've got an antibody, I'll just draw it very simply, and you've got your pathogen, then because they can bind like that, they could potentially bind to two different pathogens, and then there might be another antibody like that binding to a pathogen over here. So you've sort of clumped your pathogens together. Now, the other important thing to be aware of is the graph, which you are used to from GCSE, which is the graph of time against antibody concentration. So when you first get infected, the first thing you notice is that there is a time lag before anything happens. It's a few days. Then the antibody concentration rises. So this is a time lag in which you had to do phagocytosis, produce your APCs, activate your T helper cells, then activate your B cells, then the B cells clone, then they differentiate into plasma cells, and then they secrete antibodies. That takes a few days. So the antibodies increase in number, you fight the infection, and the antibodies drop again. Now, if you imagine then that at some point you have a second infection, what you notice is that the antibodies rise very fast, so much more rapidly, to much higher amounts, and there's no time lag at all. Okay, so there's three differences. There's no time lag, the increase is much more rapid, and it goes to higher amounts, and then it drops more slowly. And that is because of the presence of memory cells. So instead of waiting for all of that to happen, the memory B cells can straight away differentiate into plasma cells and secrete the antibodies. Okay, and that's why you have no lag. So if we put it as a sticky note here, okay, so after the second infection, there's no time lag. Antibody production is more rapid, more antibodies are produced. And this is because the memory B cells can differentiate straight away, forming plasma cells which secrete antibodies. So in terms of vaccination, the purpose of vaccination is to fool your body into doing what we call this primary immune response, i.e. make the memory cells, so that the secondary immune response will happen if you ever actually meet the infection. So what we want your body to do is to make all those memory cells. We could make your body do it naturally, but some of those infections might be very, very dangerous. So the purpose of vaccinations is simply to produce memory cells. And there are different ways you can do it, and some are better than others. So you can just inject the antigen, and it's effective, but the antigen is just a molecule. It doesn't behave like a living cell. You can inject a dead pathogen. The worry with that is that if any of them are still alive, they could cause disease. You can inject what we call an attenuated pathogen, which means one that has been made harmless. Again, the problem is that they could possibly mutate and become harmful. So these two you wouldn't do with a particularly dangerous fatal disease, just in case something goes wrong. You're more likely to use the antigen or as they've used in the current COVID, the use of messenger RNA, which can be made by the cells to produce antigen in process of transcription and translation. Now, the only other thing to know about vaccinations is the idea that we can split it up into what we call active and passive. So active immunity, as we call it, active immunity is when your body actually has to produce an immune response, i.e. produce antibodies. Passive is when you're given them. So if we'll deal with the passive first, it could be natural. Okay, so an example of when you're given natural antibodies is across the placenta. Or it could be artificial, which is when you're given antibodies. So for example, if you get caught, get cut by a saw, for example, you'll be given a tetanus shot, which will contain antibodies. In the case of active immunity, you're making your own antibodies. So again, this could be natural, i.e. you get the disease, or it could be artificial in terms of, we're talking there about vaccination. Okay, I will talk about things like HIV, ELISA, monoclonal antibodies in the next video.